Hi dear students, a warm good morning to all of you. And today we're going to talk about a specialized staining technique called the Fuelgen staining. Fuelgen stain is a staining technique which was been discovered by Robert Fuelgen as a term like it was named after its discoverer Robert Fuelgen in 1924. And this is a technique which has been used to identify DNA in cell specimens, especially when the histological analysis of cells are being carried out in order to identify the presence of chromosomal material or DNA in these cell specimens, we use the Fuelgen stain. The process of Fuelgen staining, it mainly depends upon the acid hydrolysis of DNA. So usually we do not use strong fixative agents when we are using a Fuelgen staining. Instead, we use normal or light acids, not very high acids for the staining technique. Now, how does a Fuelgen stain look? That's the next question. Let's explore that. Now, this is a Fuelgen stain which is being carried out of a human sperm and a buccal tissue. You can see that the background is considered to be as white, and where all the genetic material, this is the cell, this is a buccal tissue, and this is a sperm cell, and within that where all there is DNA, okay, it has been present as a purplish or a magenta color is being obtained. And we can also see in the at some areas, the intensity of the color is also being found to be different. So it indicates the amount of the DNA also being present at that particular area. Now, what is the principle of this staining technique? That's what we need to explore next. But before going to the principle of how Fuelgen stain enables you to see DNA, I need to just say the basics of the structure of DNA. As we all know, DNA or are deoxyribonucleotides, and they mainly consist of a base, a sugar, as well as a phosphate. And the base can be a furan or a pyrimidine. And purines, they mainly include adenine and guanine. And the other ones, the pyrimidines, it mainly includes the thymine, cytosine, and uracil. Now, the sugar in all the nucleotides, it can either be ribose or deoxyribose. But when we talk about DNA, of course, we know that the sugar which has been present in it is deoxyribose, and the bases which have been present in it are adenine, thymine, guanine, and cytosine. But uracil is not being present. So, when a base and sugar combines, we call it as a nucleoside. And when this nucleoside combines with a phosphate, it is called as a nucleotide. And when various polymers of nucleotides combine together, we get a DNA polymer. So this is the basic or structural components of a DNA. Now, as I told you, if you go deep into the structure, you can see that this is this is a structure of the pentose sugar. Now, the pentose sugar is here, and this sugar is combined with a base, either purine or a pyrimidine base, and at one end we have the phosphate. So these are the three components of a nucleic acid, and if you go to replace them with the bases, you can see that it is either adenine, thymine, guanine, or cytosine in the case of DNA. Now, how does Fuelgen staining enable you to see your DNA? That's what we are going to see. During the process of Fuelgen staining, it mainly consists of, there are different modifications of this technique, but the basic thing, it includes two steps. The first step is to treat the cytologically fixed material. Of course, suppose you have tissue specimens that we take here, so you will fix them with some techniques like either formalin and all that mild fixative agents are being used, and then after fixating, these are being treated with one normal HCl in a water bath or oven at 60 degrees centigrade for 80 to 10 minutes. This is mainly to carry out the acid hydrolysis of the specimen. I guess you remember what is fixation and what is the purpose of fixation, as we had mentioned in the previous staining techniques also. But here in Fuelgen staining, we are using mainly a chemical fixation, which has been carried out by formalin. And after that fixation, that specimen is undergoing acid hydrolysis, which has been carried out with one normal HCl in a water bath or in an oven at 60 degrees centigrade. Now, afterwards, the material is immediately transferred into Schiff's reagent at room temperature for at least 30 minutes or until the tissue stains deep purple. And after 30 minutes or when the tissue specimen becomes deep purple, you wash it with running water for about 5 minutes and rinse it in distilled water. And sometimes you can view it as such like that, so that what the background is not being stained. And in some cases, what you do is you can also counter stain it with certain other stains like light green SF, yellowish stain, and all that. That is an optional thing. So, but what happens is all those regions where DNA is being present, it will take up the first stain or the Schiff's reagent, and it will appear to be purple in color. This is the protocol. But now, how does Schiff's reagent selectively enable you for DNA staining? Now, previously we had mentioned about the what Schiff's reagent is the one which will is being used. But before the Schiff's reagent is being added, we also do a process called acid hydrolysis for the DNA molecule. And when the acid hydrolysis is being carried out, now this is a DNA molecule, and what happens? The base is removed from the DNA molecule by the process of acid hydrolysis. And as a result, now this is a glycosidic bond which is forming between the sugar as this is the sugar and this is the base. This glycosidic bond, which is between the sugar, that is deoxyribose, and the base attached to the DNA, gets broken with the help of acid hydrolysis. And that results in the formation of an aldehyde. Okay, now once the aldehyde has been formed, this aldehyde can be detected with the presence of Schiff's reagent. The aldehyde formed after acid hydrolysis combines with Schiff's reagent and results in the formation of purple color. So that's the reason how you are able to visualize DNA when you stain it with Schiff's reagent after acid hydrolysis. If you do not do acid hydrolysis during the staining technique, aldehydes will not be formed, then the detection of DNA would not be possible. So in the protocol, we need to follow the step of acid hydrolysis using mild acids, and after that you have to treat them with Schiff's reagent so that those regions where the or when those regions where the DNA is getting what acid hydrolyzed because of the removal of nitrogenous bases, they will form aldehydes, and those aldehydes react with Schiff's reagent, resulting in the formation of purple color. So you can interpret your result like this. DNA will appear to be red or purple or magenta in color. Nuclei will appear to be red or purple or magenta in color. And if you are staining it with the counter stain, the counter stain will appear to be green in color. And it is recommended that the material to be analyzed should be done on the same day. But if you want, you can keep it at 4 degrees centigrade for several days also if you want. And same as we had mentioned, the acid hydrolysis creates aldehydes, and the aldehydes react with Schiff's reagent, which results in the purple staining. And one thing which we should note here is RNA is not being hydrolyzed by HCl treatment, and thus this reaction is considered to be as DNA specific. If you go to take a particular cell, you cannot exclude it out of to have only DNA or to contain only RNA, because always in a living cell, the process of transcription and translation and DNA synthesis all happen simultaneously. And just like we have DNA within our cell, the DNA is also being converted into RNA. So the DNA and RNA are present simultaneously within a cell. But when we do the process of acid hydrolysis, only DNA gets converted into RNA, into an aldehyde, because RNA is not getting hydrolyzed by the HCl treatment. So the Fuelgen staining can be exclusively used for the detection of DNA. Now in this slide, you can explore DNA which is being stained in different types of cells. Now the three photos like A, B and C, they mainly refers towards the ordinary staining microscopic views of Fuelgen stained chromatin, and D is a fluorescence microscopic staining image of the same of another image. Okay, it's a Malpighian tubule of Triatoma infestans. So what is, now you can see in A is a mouse hepatocyte. Hepatocyte is in liver cells. So in the liver cells, now background staining is not being done in A, B and C. It's just the Fuelgen staining alone without counter staining using second dye is not being done. And so A is a mouse hepatocyte. You can see where there is DNA, they're all you can get a purplish color. And if you go to look into the second one, it is a caco-intestinal cell. Now what is a caco-intestinal cell? A caco-intestinal cell is a it is a type of cancerous cell, cell culture based cell, where you can see that some regions there are small dots like which are being found to be clustered together, and these mainly represents the apoptotic bodies which have been present within the cell. So basically you understand that where all that DNA is being present, the Fuelgen stain is being taken up. And if you go to look into the C is a Malpighian tubule, and the nucleus of Triatoma infestans, which mainly causes Chagas disease and all that. No, Triatoma infestant is a bug. Not a bug, it's a vector like which transfers the disease. And so in the Malpighian tubule, you can see the nucleus. See, you can see within the nucleus, of course, we know the genetic material has been found to be greater than the cytoplasm. So the location of or the localized identification of the nucleus of the particular cell is also been possible based upon the intensity of the Fuelgen staining at certain regions. That is, when it has been found to be more at a particular region, you will be able to see the what? The presence of nucleus. That is, now here heterochromatin or the thickened area of chromatin can be found here in this particular image. Now this is the D is a fluorescence based imaging. I'm not explaining into that because that is not the uh, core area of today's talk. So let me just give an outline. You can either for Fuelgen staining, you have to fix your tissue, then you have to rinse it, hydrolyze it with acid, and wash it again, stain it with Schiff's reagent, and sometimes people give a bisulfite treatment, then give running water treatment, air dry and view it. Now, what is the basis of each and everything? Fixation, of course, dehydrates the cell, cross-links the nuclear proteins and prevents the loss of DNA. When you run it with tap water, it removes all the fixatives. Then acid hydrolysis depurinates DNA and generates free aldehydes. And then you give a short rinse with a weak acid to remove the Schiff's reagent. Stain it with Schiff's reagent, then bind the Schiff's reagent binds it. Okay, now here, sorry, uh, sorry, in the previous rinse with a weak acid, we are mainly trying to prevent the carryover of strong acid to Schiff's reagent, because strong acid is not needed for the Schiff's reagent. So that's what we are just going to put the Schiff's reagent in the next step. So to prevent the association of strong acids with the Schiff's reagent, you give a wash, and after the wash you give a Schiff's reagent. Then sometimes they also wash it with bisulfite water. uh, Bisulfite water is a decolorizer which will remove the unbound stain. Okay, and then tap water rinse is being given, and you wash them and allow them to dry. Okay, and then you view it under the microscope. This is the basic principle on the outline of the Schiff's reagent. And something which I would like to extend upon the Fuelgen staining is that it can be used to evaluate various DNA damages, and at the same time you can also combine Fuelgen staining with some instruments such as a microdensitometer or a microspectrophotometer, and semi-quantitatively also you can estimate the amount of DNA which has been present. So like sometimes you can say if the intensity of the pink or the Fuelgen stain has been found to be more in one particular organelle, that can be estimated. Or at the same time, the different cell cycles, different stages of the cell cycle. Now sometimes the DNA will become diploid, sometimes the DNA will become tetraploid and all that. So to depict all that, you can use Fuelgen staining also quantitatively, semi-quantitatively specifically speaking, and you can estimate the amount of DNA in the tissues also when you combine Fuelgen staining along with microspectrophotometer also. So with this we come to an end of Fuelgen staining. Thank you.